from the plant that said, here's how many pounds or ounces of waste material they picked up off the floor, and to put it in a spreadsheet. At the end of the summer, give them a number. Super boring, right? Like it took, I don't know, maybe like 0.5% of my brain power. So what I learned from that one was basically how to get up and go to work every morning, right? How to get up reliably, go someplace, be there on time, do what I was supposed to do, engage, and then leave. Didn't really learn anything else. I also learned every word to the Trisha Yearwood song, She's in Love with the Boy. I won't tell you why, but if you ask me during the Q&A, I'll give you more. So the summer after my sophomore year is when I got my first job that was more of a real engineering job. And I went into the pharmaceutical industry, worked at a company called Merck in the manufacturing division. And this was fantastic because I had developed an interest in the biological sciences and that part of chemical engineering from some of the information that Dr. Cargill had given to me. And this was pharma. It was actually vaccine manufacturing. So it involved a lot of biology and biological sciences. And I got really excited about the opportunity to work in an industry where I could see that what they were doing was important. It was about delivering medicines. And Merck has this slogan that says, medicine is for the people, not for the profits. If we remember that, the profits will follow. And so I felt very inspired to be in this company. The other thing I learned, though, was that I could get a PhD and I didn't have to necessarily become an academic, right? Like I knew all of my professors had PhDs, but I hadn't actually been exposed to people who had PhDs, which is a research-based degree, who weren't doing more of in a research enterprise. And so this sort of prompted me to keep thinking about advancing my education because I realized for the first time that I had a lot more options for what I could do with a PhD than I had before. My third job was more traditional kind of process engineering. And again, I don't have the time to go over the details in this part of it, but this was chemicals, so not pharmaceuticals, but a different kind of chemicals in the first job that I did. And what I really realized there was I didn't like traditional process engineering. I liked things that were more on the cutting edge, more discovery-based, rather than can we just keep this manufacturing facility going? I also had like a epiphany in that one. Again, you can ask me about it later if you want to, that like really crystallized for me that I was gonna go to graduate school. So that was after my senior year, I applied to graduate schools. And then the job that I had after senior year and before I started grad school was back in Texas at Exxon Production Research, working in the oil and gas field and doing computational research. And I learned there that I didn't like that. To me, the highlight of that job, about a weekend, I went and bought a calendar. And every day at the end of the day, I would just put an X through one more day that was gone. And like, if you're doing that, like you're not loving your job, right? And the overall area, the goal of the work I thought was interesting, but I didn't like just sitting at a computer every day. I knew then when I went to graduate school, I was very much interested in working as an experimentalist, meaning working with my hands, getting into the lab, mixing things together, making measurements, drawing conclusions from those measurements, rather than building models and drawing conclusions from the models. I went to grad school at Berkeley in California. I'll just say very briefly, a big motivation for me going to California was that I was cold. I had grown up in Texas. I came here. My senior year was like the coldest winter that Massachusetts had had in quite a while. And I was like, you know what? I'm ready to leave winter behind. Berkeley was also a great school. Okay, so my sole motivation wasn't the weather, but it certainly didn't hurt that I could go from sub-zero degree weather out to Northern California. So for my PhD, I worked with a guy named Jay Kiesling, who's actually very well known in the field now and had just gotten started when I was a graduate student in this area called metabolic engineering. So I have a few slides to just kind of give you some context for what I mean when I say metabolic engineering. I have here a couple of products that you should not be familiar with at all. But the point here is to say that these products are the result of sugar being converted into ethanol by a microbe, by a yeast. Okay, so we know that the yeast is going to take sugar and convert it into ethanol. There are very specific chemical structures that are associated with that. The sugar is predominantly glucose. It's a very specific molecule, and the ethanol itself is a specific molecule. And so you guys have taken chemistry, you know there must be some chemical reaction that describes how you go from the substrate to the product or from the reagent to the product. So this is the overall reaction of biology. It takes a molecule of glucose, you take some ADP, you can produce ATP, you produce CO2, you also produce ethanol. And those reactions happen because of proteins, specialized proteins that are enzymes. Those enzymes are encoded by DNA because DNA goes to RNA, RNA goes to protein. So what we're able to do then in metabolic engineering is take genes, these specific pieces of DNA, introduce them into a cell to make enzymes, specialized proteins that will do these kinds of chemical reactions. And of course, if we're talking about something like a cell taking a sugar molecule and converting it to ethanol, it's not just one molecule or one enzyme that does that, but a lot of different enzymes are involved in this process of going from a simple molecule like sugar to ethanol. Now, I've described this to you as if you were working in a test tube and you put in sugar and you added in some enzymes and then you got product at the other end. In fact, what you have is a cell that's doing this, that's taking the sugar, and it's not just taking that sugar and going into ethanol, but it's taking that sugar and passing it through a lot of other chemical reactions. They're still reactions, but they're encoded now by these specialized proteins that are enzymes. And they're making many, 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 many hundreds of molecules participating in many thousands of reactions. This is not news. Biology as chemistry, meaning biology with this ability to do chemistry, has been exploited for things like fermentation in food, but also to make products that are useful to humans. And that includes drugs. So what's shown here is a compound called lovastatin, which was one of the first cholesterol-lowering drugs. It's a natural product normally produced by a fungal organism. What's on the other side is a compound called cansitis, which is an antifungal, also produced by a biological compound. That's for therapeutics, but it also turns out that you can use these cells to make amino acids. You can use them to make other organic acids that are made at very large scale. So what metabolic engineering really is about is taking this context of biology, doing chemistry, and asking, how do we actually improve upon the ability to do that? How do we make more products? How do we make products more efficiently? How do we make them more robustly? In large part so that we can actually expand the scope of compounds that are available to us for medicines, for materials, for industrial chemicals, for whatever you might imagine. So I'll just quickly say, the examples I gave you on the other side were really about if you have a cell and it can make those things, how do you actually have that happen better? But there are a couple of examples that I give here of cases where these are compounds that are produced in one type of biological system. And then what engineers have done is figure out how to make something much simpler, these E. coli cells, produce the compound instead. So in the case of this compound called 1,3-propane-diol, it's naturally produced in this bug. You can see the name there. Yes, the pneumoniae is for pneumonia. That's a compound that organism that causes pneumonia. So you don't want to grow it up in very large reactors to make this compound. Instead, what engineers were able to do is to figure out how to engineer E. coli, a very simple bacterium to do that. And then artemisinic acid is actually native to a plant. And there were ways to figure out, engineers rather figured out how to transport those pathways or those enzymes that make that compound from the plant into the microbial cell. So when I was in graduate school, this is what I learned how to do. My work was really focused on how do we facilitate this process of engineering cells to produce a number of these different compounds. When I finished graduate school, I actually knew I wanted to be a professor, but I also wanted to have some experience in industry. So I have what I describe here as my first real job. It's in quotes because it turns out like this job is pretty real and it has been for a while, right? But out in the non-academic world, this was at a pharmaceutical company called Merck, at Merck Research Labs. I was there for 4 years, working in a number of different areas. And I just want to highlight biocatalysis because it really became the segue to most of the work that happens in my lab now. And so biocatalysis is using enzymes, specialized proteins that do catalysis to do chemical reactions. You can do this with those enzymes being purified. So again, mixing them up in a test tube, as it will, or you can think of it the same way we think of those yeast cells, taking sugar and making ethanol by having a whole cell do these kinds of conversions. When I was at Merck, all the projects we had for biocatalysis were about doing single reactions, right? So as an example of that, here's a slide actually from time I spent at Merck where this enzyme that's shown here, PHDH, stands for phenylalanine dehydrogenase. What it normally will do is make the amino acid phenylalanine, shown on the right-hand side, from a precursor. But there were researchers at a pharmaceutical company called Bristol Myers Squibb that figured out, well, if we can get the enzyme that does that reaction to work on a different kind of molecule, then maybe the fact that they're close enough means that I can convert this molecule here to the amine group. You can see here's where the reaction is taking place by using this same enzyme, even though those compounds are a little bit different. So the way that I always summarize this is that biocatalysis allows you to take an unnatural substrate, so something that the cell wouldn't normally see, and by doing that, it actually allows you to make new kinds of products. So I spent time at Merck kind of working on these single step reactions. I had spent time in graduate school where I had learned how to engineer whole cells. And so the question that I had was, well, could you actually, rather than being focused on pathways that already exist, can you find a number of these cases of enzymes working on these new substrates to put them together and to make entirely new kinds of pathways to get to new kinds of molecules? And it was really with that sort of framework that I started my research here at MIT. So I came to MIT, it'll be 20 years ago in about 3.5 weeks, to start my research lab here. And the primary motivation and driver behind everything that we do is to go from a world where we depend upon fossil inputs, so petroleum, to get fuels and chemicals, to a world where we can depend upon biomass, upon renewable, sustainable substrates to be able to take simple inputs and make the products that we want, with a particular focus on chemicals rather than fuels. So for fuels, we can think about batteries, electrification. But if we're talking about actually making materials that are useful and that traditionally come from oil and gas, we need different ways to be able to do that. So it's been about 10 years ago now, I did this exercise where I took all of the titles of the papers that we had published and put them in. I just realized that. So back then it was called Wordle. Wordle is something different now. Who plays Wordle every day? You guys got today's? It's a good one. I do it every morning. I do the New York Times Mini and then Wordle and then the new Strands game. I do connections at night with my daughter. It's a bonding activity. So the old view of Wordle, well, word cloud is what Wordle was. So you put everything in and see what dominates. So when I did that, here's the image that was generated. And it's fantastic because what pulls out is production. You get I forgot there was animation. You get engineering as a keyword there. E. coli pops up. And then the other one that you can see pretty predominantly is pathway. So if you take those 4 phrases and ask, well, what does that really mean? What we do in our lab is to engineer new pathways, mostly in E. coli, for the production of chemical compounds. Okay? And I describe that as teaching old bacteria new tricks, right? So essentially what we do is to say, we've got the cell normally can make a lot of chemical compounds. And what we're trying to do is to figure out how do we get more compounds to be produced based on specific needs. So as one example for this compound we've made called glucaric acid, it takes 3 genes or 3 pieces of DNA from 3 different organisms. One is a yeast, one is actually a mouse, one is a bacterium. There's another compound we've made that takes 4 genes from 3 organisms. The one we've done that's most complex has required 10 genes from 8 different organisms. And in all cases, what we're trying to do is to figure out how do we make new kinds of molecules, and with a particular focus on having them be economical and environmentally responsible. So reducing the footprint that's associated with that. I have also been involved in a startup that's looked to commercialize some of the work out of my lab. And I'll give one example for this compound glucaric acid for what that's being used for. And the example here is with cooling towers. So you guys probably have not been to a chemical plant. Hopefully you haven't been to a chemical plant. It's usually not a place that high school students hang out. But in any large manufacturing facility, water is a big deal. And you have different parts of the facility where you need to heat things up, you need to cool them down. And the water that's engaged in that has to go through these different systems and much of it ends up in what's called a cooling tower, where the water comes in at the top. And actually just the process of evaporation, as you let it fall from a height down to lower, the temperature drops. So as you evaporate water, you remove energy from it, and the temperature of the resulting water goes down. That water has to actually be treated before you can discharge it to the environment. And so traditionally, the way you do that is by adding a compound called phosphate. The problem with phosphate is that when too much of it shows up in natural waterways, it's food for algae. And as the algae grow, they block out the light, and they also consume all the oxygen that's available. And so this process ends up being called eutrophication. If you have an area where eutrophication has occurred, a lot of the fish and other aquatic wildlife dies because the algal bloom actually kills them. So we knew for a while, actually, that glucaric acid was a great replacement for phosphate. It's naturally degradable. It doesn't cause any of these eutrophication problems. And so that's where we had really been pushing the utilization of this compound. What we actually found out was that by using this compound to treat the water rather than the phosphates, it actually helps the performance of what are called heat exchangers. So these big pieces of equipment you pass the water through. It helps those such that not only do you help the environment, but you can actually have more efficient operation, reducing now CO2 emissions, and then also saving large amounts of money. I'm going to quickly give two other examples of work we're doing as collaborations. This is with, as part of the MIT Climate Grand Challenges project, and it is with Chris Voigt as the lead PI. And the main point of this pie chart is to show that agriculture, ironically, has a very high CO2 footprint. We usually don't think about that because we think about plants as pulling CO2 out of the atmosphere, and that's a good thing. But the problem is that if you have a high requirement for fertilizer, the process of making fertilizer is very energy demanding as well. So the idea that Chris and others came up with is that there are certain bacteria that naturally associate with roots of plants that are able to take nitrogen out of the atmosphere and turn it into ammonia fertilizer. The problem is that the crops that normally do that are not really crops that are designed to feed the planet. So they're not cereal crops, for example. So what we're working on is trying to figure out, could you actually engineer these cereal crops to be able to now have bacteria associated with them that would fix nitrogen? And then the sort of holy grail, this is, there's a company that Chris Voigt co-founded called Pivot Bio that's doing that. And then the holy grail is actually being able to engineer the crops directly to be able to pull nitrogen out of the air and make their own fertilizer. And the last example I'll give you is around plastics. You can see the numbers that are there, but they shouldn't be surprising to you that plastics are ubiquitous. They're everywhere. They are very long lasting, and so they cause problems with contamination for long periods of time. And they break down into microplastics. If you guys saw Happy Feet, you guys remember the movie Happy Feet? Yeah, remember like the Grand Poobah actually had like a soda thing around his neck, and they thought he was really special, but it was really very horrifying and sad to look at. I don't know if kids found it as horrifying and sad as I did as an adult, right? So the issue is well known with plastics. The problem is that we actually need plastic. So it's not just convenient. In some cases, it is convenient, but it's not just convenience. It's actually, there are places where plastics have a dramatic influence on quality of life. Food preservation is a great example of this. So I've been involved in a collaboration. You can see my collaborators on the bottom for a while with this idea of being able to design materials for degradation. And so we start from asking, how do we actually make the building blocks of plastics from things that are renewable? So how do we take our bacteria, engineer them to make the building blocks? But then after you have those building blocks, how do you make sure that they can actually be degraded in the environment so they won't persist? So we've been working on developing new kinds of methods so that that involve robotic screening, so that you can reduce the time to evaluate biodegradation from several months rather, to just a couple of weeks. And then we're also working on how do we actually change these assays to be more reflective of what the environment that the plastics will go into actually looks like. So rather than looking at a single microbe being able to degrade plastics, what happens if you look at a collection of microbes that might be found available in soil? So I went through things pretty quickly. I just wanted to give you some highlights. I'm just going to end by acknowledging the folks that funded the work that I've talked about, and acknowledging the people who are in my research group. And this is my favorite picture that was actually produced by MIT News some years ago. That to me is the perfect representation of what we're really trying to do, which is how do we take these bacteria, recognize that they are really just teeny tiny chemical factories, those little smokestacks or CO2 coming out as the cells are growing. But really, how do we switch our thinking from relying upon non-renewable fossil-based feedstocks towards being able to exploit and leverage renewable, sustainable biomass-based feedstocks to make a wide variety of chemicals and materials. So with that, I will say thank you, and then I'll move in the center and take some questions. Yes.
So for biology, there's this thing where you can see how far things evolved since they were first created. And E. coli had the longest. It was not even funny, like even stopped very short. And you can just see how much E. coli is. What do you think the most interesting thing about E. coli is?
Okay, so I'm going to come back to that in a second. Tell me your name and where you're from.
My name is William.
From Newton? Okay, William from Newton. So William said, in biology, you can tell essentially evolutionary lifestyle or lifespan. So you can see how much things have evolved from a certain point in time. In humans, evolution is fairly slow, but in something like E. coli, it's evolved a lot. So then the question was, what's my favorite thing about E. coli? Is that right? So my favorite thing about E. coli, it's very easy to answer. It is very easy to engineer. And it has been worked on for so long. We have a lot of tools that are available to do really fun and interesting things with it. So one of the questions I get asked a lot is, why do we work in E. coli when we're trying to make some compounds that may not be very good for E. coli to produce? So for example, if we try to get E. coli to make ethanol, it can't make nearly as much ethanol as yeast can before the cells just die. And my response to that was always, E. coli is a great prototyping organism because so many people have worked with it for such a long period of time. There are a lot of tools that are available. And so we can explore a lot of different configurations. We can explore different ways of, or different types of tools that we can build. And we can explore, in our case, a number of different versions of pathways to figure out what's feasible and what's not feasible. And because of the speed that that affords us, we get a lot of information very quickly. It does have limitations though. And so we have some work where we're looking at non-E. coli based organisms, but the timeline it takes to do an experiment in those is honestly about 6 to 10 times longer than it takes to do it into E. coli. So the speed with which we can do work in E. coli is by far my favorite thing about the organism. Yeah. Tell me your name and where you're from.
Hello, I'm Elliot, and I'm from Southern California. You said that E. coli was really good for prototyping. Is the end goal of your research to be able to implement these processes with E. coli, or is it to explore options for use in other organisms?
Yeah, it's both. So there are some cases. I mentioned the startup company I'm involved with, Calion. Our commercial process for that involves E. coli. So in that particular case, the goal is E. coli, and we're happy with E. coli. We have another project that we're working on where we have a completely different organism, and the ultimate goal is to be able to use that organism for production. And in that case, we've already done all the prototyping in E. coli, and now we're trying to move it to this other organism. But we also have to develop the tools to engineer it as we go, because it's not as well developed as E. coli is. Yeah. Here.
Here, I'm on this side. I'm faster.
And then you'll pass it about, actually, we'll do in front. Yep. And then you'll pass it behind you when you're done. Yeah. Okay.
My name is Isaac, and I'm from Houston, Texas. I remember you were talking about how your first job, the super boring one, was just putting numbers in a spreadsheet every day.
Yes.
What was the result of that? Like, did they decide to throw the material away, or did they decide to recycle it?
I have no idea. They never actually told me, which is the other thing about it, right? I spent all this time doing mindless work and I have no idea what the impact was on it. And will say I have wondered since then, not only what the answer was then, but what the answer might be today, right? Because it all depends on what the value is of that compound. And polypropylene is the largest volume plastic in the world. And manufacturers have been consolidating. And so prices have fluctuated a lot for it. Prices during the pandemic went way high because supply chains were disrupted. And so if you were working in the plant and I don't know, August of 2020, it probably was worth it to recycle those pellets no matter the cost because you just didn't have access to materials from there. But I don't know what the outcome of was it. And I don't know anybody who works there now to be able to ask.
Okay, sorry.
Hi, my name is Ria and I'm from Southern California. And I'm really interested in plastics. When I was a little kid, I was always told that plastics will stay forever. And so I had a question for you in terms of, do you think that we can use chemical engineering in the future to potentially like destroy the current plastics that are on this planet and try to repurpose them so they're not polluting our environment?
Yeah. So absolutely. And there's a lot of work being done, not just in chemical engineering here at MIT, but in ChemE departments all over the country and in many parts of the world looking at this. I'll tell you what the challenges are that are associated with it. And there are a couple of them. So let's take something easy first. Polyesters. Polyesters are actually relatively easy because the linkages between them, the ester bonds, are ones that are pretty easily broken. So there are even bacterial species that can degrade certain forms of polyesters. Contrast that with what's called polyethylene. Polyethylene is just a long string of carbon and hydrogen, and it's sort of difficult to break into it. So the biggest challenge that's associated, there's two big challenges associated right now with recycling. One is the cost. Actually, 1 through 5 would be the cost, because the second one is around the heterogeneity. So if you think about what you start with when you make plastics are these really precisely defined and very clean monomers, these individual chemicals that you mix together. If you don't have a clean way to break them down, what you end up with when you try to now decompose them is this mixture of, in the case of polyethylene, that's a 2-carbon molecule. So you get some 2 carbons, but you get some 6 carbons, and you get some 12 carbons, and you get some 16 carbons. So you have to clean that up. And that's also chemical engineering, to be able to separate those different fractions. And it's not that it can't be done, but today, by the time you've gone through that whole process, the cost of now having a recycled material is significantly more expensive than taking something new. And I can't remember what the multiple is right now. And people aren't willing to pay for that. And some people can't afford to pay for it, but they're not willing to pay for it. I bought, about a year ago, for the first time, I was in Target. I needed aluminum foil, and Reynolds, which is a big aluminum maker, was selling aluminum foil from recycled aluminum. And I bought it because, I have a good job. It was not cheap, right? So it was about 20% more expensive than the regular for about 60% of the amount. So it was a smaller box that cost more money, but I sort of figured I was willing to do it. I had to buy aluminum foil again on Saturday, and I didn't even see it in the store. So I don't know if that store just wasn't carrying it, but what has happened from time to time is that you'll see products that pop up that are recycled, they're from renewable resources, but they're more expensive. And because they're more expensive, consumers don't adopt them. So there's some technical challenges in terms of getting everything where it could be nice and clean to the point where you could use it in the same way. But those technical challenges come with economic challenges as well. And then that's when you end up with like sociology and consumer psychology and all these kinds of things that come into play. But for certain, there are chemical engineers working on it, both for biological processing and for chemical remediation as well.
Hi, I'm Laura, and I'm from around the Boston area. This is a question about kind of your process in your work. I guess when you're mixing multiple chemicals together, how much of like educated guess do you know? Like, do you know if have a guess about, oh, these two chemicals would work together, or are you just like throwing random things and see what it works?
Yeah, so this is a great question. So in our case, we're typically not mixing chemicals together. When I say chemical, I'm thinking about small molecules. We are mixing though proteins together and asking, can they do specific reactions in order to make new chemicals for us? And in that case, it definitely is an educated guess, right? Meaning that we have evidence and from previous work, so evidence and published literature about a specific reaction that we know an enzyme will do. So we don't have any question about whether or not the enzyme will do the reaction. We often have questions about whether or not it will do that reaction on a new molecule. And oftentimes what we're looking at is making something that's new. So we need to start with new reagents for that. And because, to your point, we don't know exactly if it works or not, we take what we would describe as a library-based approach. So if we're looking at a particular reaction and we think there's an enzyme that will do this reaction step, we don't just look at one version of that enzyme. We put together a panel of anywhere from 6 to 12 of those enzymes in order to test them and see if we get any kind of activity. And then based on the results of that, that might guide us towards looking in other places to go even further and make sure, or to see how high we can push the productivity there. But we certainly have to rely upon what's published in literature. It's not completely blind for certain, but we can't be positive that just one thing will be okay. And so we try to pull different variations. Homologs is the word that would be used for. We pull different protein homologs together to try to find at least one that will give us the activity we're looking for.
We're going to balance off the in-person with some of the people on Zoom.
Okay.
So we have people on Zoom. And the one big question from the Zoom group was, what about this song? People want to know the story behind it, and how do you know every word?
Okay, let me tell you the story behind the song. So imagine a world before cell phones. I know it's impossible, but just imagine for a minute a world before cell phones. Do you guys know what an answering machine is? Okay, so anybody not know what an answering machine is? Hold your hand high and say, I am young and I've never seen an answering machine. Okay, so an answering machine, back in the olden days, an answering machine was a device that you would physically plug with a wire your telephone into. And if you did not answer the phone, the machine would answer the phone. So it would close the circuit and it would have an outgoing message that said, hello, my name is Prather. I'm sorry, I've stepped away from my desk. Please leave a message. And it would beep and you would have a message the same way as you do on your cell phones now. But in these olden days, in the days of the summer of 1991, now I have to introduce something else, cassette tape. You guys know what a cassette tape is? Okay, no cassette tape? Oh, you do? Okay. So in the olden days, the answering machine had a cassette tape in it. And on one side, it would record the outgoing message. On the other side, or the message you would say, the other side, the incoming message. And then when they got really fancy, the outgoing message was embedded in a chip, but you still had to have the cassette tape to record the outside. Well, it was just a regular cassette tape. And so I shared an office with my boss, who was a huge Trisha Yearwood fan. And Trisha Yearwood had an album that came out that summer, and she bought a cassette tape. And every single day in the office, she would take out the answering machine tape because she was there. She didn't need an answering machine. And she would put in the Trisha Yearwood tape, and she played that song every single day for what amounted to about half of my internship, so about 6 or 7 weeks. And so I had no choice but to learn every single word to she's in love with the boy, no. She's in love with the boy, no. She's in love with the boy, no. And even if she has to run away, she's gonna marry that boy someday. So that's how I know every word to she's in love with the boy.
All right, we're gonna go to, one more question from online.
Yes.
And then we're gonna keep it, so we'll send you all the questions online.
Okay.
Via email for later, so you can take a look at them
Okay.
as well. But one more question here. You mentioned scaling up processes from lab level to industrial level.
Yes.
What strategies do you use to make sure that those lab level successes can be replicated at industrial level?
Yeah, that is a great question. So in our research lab, one of the big things that we try to do is if we're creating something that's new, the lab level for us often involves a scale that's anywhere from 1 milliliter to 50 milliliters, right? And so those are in like a flask or a test tube that doesn't really mimic at all what you may find if you went into commercial scale. But we also have reactors, actual instrumented bioreactors in our lab. Those operate at about 2 to 3 liters, which doesn't sound like a lot, but the fact that we have all the instrumentation that goes along with it, for the most part, if you can get a process to work at that level, you have significant confidence that you can get it to scale up. So what we actually do to try to ensure that what we do is scalable is to, we don't do it for everything, but for certain new tools that we develop, after we've tested out on a small scale, we actually make sure we can replicate it in this larger scale bioreactor. And if we can't, it tells us that there is some challenge there with being able to take what we've learned at the small scale and put it into larger scale. And so we go back to the drawing board and try to figure out where the disconnect is there.
All right, super. And Kevin, if we can have Ishita and Yubo on the screen for our virtual t-shirt presentation. There we go.
Hi, I'm Ishita from Medlytics. Thank you so much for your presentation today. I especially love the part where you explained how you were tortured by that song over and over again. And just as a thank you from all of us, including the Zoom population, we would love to virtually present you with a ViewerWorks t-shirt. So I'm going to hand it to you.
Oh, there you go.
Thanks, Ishita. I'm Yubo, a rising senior from Lawrenceville, Georgia, and a Medlytics program student. I'm passionate about biomedical science and the application of computational biology method, and it's truly inspiring to hear about your research and the career path. We would like to thank you with our BWSI t-shirts.
Well, oh, I'm wrong way. There we go. There's this way. Well, thank you both very much.
All right, super. Just a couple of quick announcements. You got the heart.
I like that. That's very nice.
Yeah. Just a couple of quick announcements before we send you back to class. One, there's been questions about whether students can get food during the program. If you're in the status center, you can buy food at that Forbes Cafe, but you should not be leaving your TAs. You should not be leaving your instructors or groups to go outside of that area to buy lunch or to get other food. You can certainly do it before the program or after the program, but while you're getting lunch, you should stick with your program. The other thing I want to mention is everyone should have their lanyards on. We want to see everybody's lanyards on because it helps identify you as part of this program. And you should be proud of being part of this program. Was there anything else, Sigrid? Bob? But
like, can we take some pictures of you with the t-shirt? And all of you will come down. Come on down.
Yeah, come on down. It can be a group shot.
Okay, here. I'll put it on. It even matches. I had a black shirt on underneath, so it matches.
Oh, I know.
That's okay. You have your lanyards on, so we know where you guys are. All right. Got it?
Thank you.
Very nice to meet you. Very
Thank
nice to
you.
meet you.
I have a question.
Yeah.
Can I ask you?
Sure.
For like, during your time working.
Because of time, we're going to start dismissing by courses. Can we have SAR leave first in the back?
Small favors.
In the back, please. Can you head out the back? Thank you. Yeah. Do we have a UAV TA here? Can we do UAV next? Head out towards the back as well.